Hi everyone. Hope you've all enjoyed the virtual talk so far at ASGCT. I really appreciate that you're tuning to this talk now. My name is Johnny Truong and I'm a part of the marketing team here at Benchling. Today, I'm going to discuss how you can be bringing life into your gene and cell therapy R&D. To give some context, I like to highlight just how much the field has progressed in the last 8 years. Starting in 2012, scientists have shown that you can reconstitute CRISPR systems for targeted, programmable gene editing. It's been an inspiration for many gene therapies today. Since then, there have been a lot of first-of-its-kind FDA approvals within the field, ranging from the first antisense oligonucleotide therapy, Kynamro, to the first gene therapy for inherited disease, Luxturna. Even in the last year, there's been a gene therapy established for spinal muscular atrophy, which has been hailed as a one-time cure, but also the most expensive treatment to date. As of today, there's likely a number of innovations that are being actively developed to combat COVID-19. But I want to pose a question for you all here today. How are we advancing the field so rapidly? My theory is researchers are changing the tools and the methods they use to get there. Researchers are developing a new type of intellect to accompany this as well. And at Benchling, we like to call this biointelligence. Biointelligence is the interplay of sample intelligence and process intelligence. You need to design, identify, and characterize samples you work with, but you should also understand how these samples were treated during experiments and downstream assays. Getting both these types of intelligence, it's difficult for any research organization. But if you're able to, you can answer questions in real time that took you maybe hours or even days. No matter what your role is, whether you're a scientist, a manager, or a director on a team, there are always insights that you can be answering. Is this the highest yielding cell line? What's the best platform for my team to use? And where are the most resources being allocated? Keeping up with biointelligence is critical for accelerating gene and cell therapies, especially as the needs of R&D have changed and will change. Whether it's to be more productive or to scale quickly, you'll need the right tools to respond appropriately. Benchling has adopted this paradigm shift, and we have grown as a company to accommodate growing needs. Founded in 2012, Benchling started with a mission to fundamentally change how research was being done. We initially built a product aimed at academics and have since expanded into many industry applications. Our first product, microbiology, is a suite of visualization and design tools on a cloud-based interface. We invested early in the potential of CRISPR research and even built a guide RNA design tool, which has become one of our mostly widely used tools today. Since then, we've weaved biointelligence into a set of products that are all unified on our platform. And this includes notebook, registry, inventory, and request and workflows. Just recently this year, we've launched two new products, Benchling for Lab Automation and Insights. Both of these products really address R&D challenges of the future. At the core and direction of our growth, it's always been listening to our users and our customers. We're really proud to say we have users at over hundreds of research universities, as well as customers in over 270 biotech and pharma companies, and all of them involved in leading gene and cell therapy research. Benchling blends a deep expertise of life science and software that helps to accelerate scientific discovery within any organization. We are proud to say we built Benchling for a variety of audiences. For academics, we give them a high degree of flexibility, a method to streamline collaboration, and best of all, it's provided for free. What you're seeing here is an example of Benchling powering a fermentation process and how it uses the Benchling notebook and microbiology to document a protocol and the construct being used. Now, both these products are very powerful on their own, with their own set of features listed here. But one of the most compelling things about it is that these products actually communicate with one another. You can embed sequences into your notebook entries, and like here, and you can even directly open out that sequence and make changes within a sequence editor. Now, we've also built Benchling to accommodate industry users. In addition to receiving the microbiology and notebook products as well, most industry scientists require other solutions to their R&D process. So let's take a look at that same fermentation process now with other Benchling applications. Our registry can keep track of all your unique samples and can accommodate unique data fields that you may want to capture within your R&D process. Inventory is where you can actually track the physical storage location of a sample that you'll need for an experiment, which is great when you're working with multiple teams in multiple locations. Requests and workflows outlines the entire workflow for a fermentation process here and shows all the steps that are involved in them. Now, if you have a liquid handler robot or any type of equipment that can use automation, you might be able to use Benchling's lab automation feature, which can execute instrument runs using our software interface. And lastly, Benchling Insights can actually visualize any type of data is data that's collected from your experiment and allow you to visualize them on this clean, nice dashboard here. Now, while I've shown you examples of what our products look like for a fermentation process, we know that gene and cell therapies are very different and very complex. Often you could be organizing a library of genetic materials or even delivery vectors, and you need to map out the relationship of constructs between one another. Also, processes need to be tailored to the type of gene editing you're doing, whether it's involving a nuclease that's cutting DNA or integration of viral DNA within a host genome. The field of gene and cell therapy is also rapidly evolving. You often need to track different experimental conditions and also changes that actually will help better characterize your product. To be more specific to this field, I like to go over a functional use case for how we've actually improved the quality of life for our users. This CRISPR gene editing workflow applies to many of the academic and industry scientists who are in this field and have also used Benchling. We've summarized a few common steps within this process. So first, you need to identify the organism and the gene sequence you like to work with. Then you need to design and score guide RNAs for a particular target gene region. Now, depending on your CRISPR system, you need to design and clone in guide RNAs and a Cas protein within a vector. Finally, you'll probably need to perform some type of experimental validation just to ensure your CRISPR system is indeed working before further downstream experiments. Now, just within these steps outlined here, we've heard a number of issues that can arise. First, sequence information can be highly unorganized sometimes within a lab. They can live in various platforms, maybe like Word, emails, or even paper notebooks. Additionally, within a lab, certain members may prefer to use different software programs, especially to design and score guide RNAs. This can often be confusing and inconsistent when you're trying to explain the results of downstream experiments. Now, all of this right now is before Benchling. What Benchling has done is to help solve these issues by standardizing intuitive tools for biology, by preserving institutional and lab research knowledge, and lastly, unifying all data and analyses across multiple experiments. Now, I want to dive a bit deeper and explain how we're actually standardizing an intuitive tool for biology in relation to CRISPR. This is an example of someone using our CRISPR tools to design guide RNAs for a specific gene. You can literally score guide RNAs within seconds while you're still looking at your target region and see where they appear on your gene. Additionally, you can export these guides and even have them auto-assembled into a desired vector for cloning. To further exemplify this, I want to show you a direct quote from our assistant professor, Ross Wilson at UC Berkeley and the Innovative Genomics Institute. He's a Jennifer Doudna alum at that too. Yeah, I'm happy to say that Benchling's CRISPR tools helped me write a grant proposal that has been successfully funded. It was very helpful to have an easy-to-use gene editing tool coupled to a user-friendly gene and protein map. Now I'm going to talk more specifically about a process that's applicable to our industry listeners here today. Among our customers, many of them have processes for viral vector production. While each company may produce viruses for very different purposes, the pipeline for viral production tends to have very similar stages. It typically starts with a team designing plasmids, which code for the outside of the virus, and then also ones that and DNA or RNA that inside the virus that it carries and delivers. You also need to work on the cell line that will be transfected with plasmids containing your virus and grow it to an enormous number of cells that will actually produce your virus. Once you have enough, you'll harvest and purify it, and then you'll likely run assays downstream to confirm the virus identity and functionality. Oftentimes, companies have a dedicated team to this process called a viral core. And here are some of the typical challenges they can face. First, samples and data that are handed off often leave out key information. This can make it very hard to find something as simple as which plasmids were used to produce the virus lot. Second, there's a lot of changes that can happen during an experiment, and sometimes many of this isn't always documented or recorded. And this can especially be true for certain purification protocols. Often scientists end up repeating these procedures without the most up-to-date conditions, which can actually affect their experiment quality. And then lastly, sometimes when you're actually communicating with other teams, a lot of the information needed to conduct a lot of quality control assays are often left out of initial email communications. This requires a lot more follow-up between teams and then also wastes time on both ends. Now, after incorporating Benchling, these companies have now been able to track their viral sample lineage back to the sequence level. They've even been able to evolve their process with full traceability end to end. And lastly, they've been able to better coordinate workflows across external teams. Now I'm going to similarly explore these two highlighted statements a little bit more. With Benchling, you can actually track all transgene edits within a sequence and trace it back to every virus prep that you may conduct. Now, additionally, you can even input unique fields that you have for a particular viral lot. If you want to capture and map relations between the transgene or even the helper plasmids that were used, and even track the exact cell line. And lastly, you can even record key data, such as maybe the titer or purity, and additionally auto-calculate properties of your virus based on its plasmid sequence. Benchling really levels up the end-to-end traceability of your process. We can actually map out your entire viral vector reduction workflow, which can be seen here. Now different teams can actually see the status of each request and communicate with one another through the product when a downstream step was completed and what the results actually were. You're able also to even track different parameters within each step and also change any last minute, and it also include any last minute changes, maybe if you use new media or even buffer conditions. They can all be reflected here. Now these achievements were really from key partnerships from our customers who are shown here. We worked together with them to better understand their problems and how we could bring about a solution. Their feedback drove us to create better products not only for them, but for all R&D processes. And honestly, we like to think that we've become an indispensable part of our customers' organizations. You might ask us what sets us apart from other solutions that you've used. First, we're built for science. We have a unified data model with built-in biological intelligence. Second, we're made for users. We're a modern, intuitive application with unprecedented user adoption. Third, we are built for IT. We have a flexible, configurable platform that keeps up to the pace with science, and we were created for insights. We want to be the single source of truth for a team, an experiment, or even program-level decisions. And finally, we deliver results. We have an amazing customer experience team that works closely with you and your R&D organization and customizes Benchling to fit your unique processes. Finally, I want to leave you all with what Benchling has kind of been doing now in light of recent developments. We're committed to helping R&D processes for gene and cell therapies, but also to really better society. With everything going on with COVID-19, Benchling has even launched our own response in combating coronavirus. We've compiled a number of COVID-19 resources, from sequences to protocols, and have equipped and empowered scientists to do their own COVID-19 research on our platform. We also realize that the network of our users are wide and vast. We've generated channels for academic and industry users to donate supplies to local COVID-19 testing centers and healthcare facilities. Lastly, one of the biggest problems today is the accuracy and availability of testing. Benchling is hoping to tackle that challenge through our product. If you are a lab working on COVID-19 diagnostic testing, we're giving away our full platform for free. Now, I just want to thank you all for your time here at ASGCT. If you have any questions for me about anything that went over today, please feel free to reach out at johnny@benchling.com. Alternatively, come visit our website at www.benchling.com to learn more about anything that was discussed and find out how we can really help your research organization today. Take care and stay safe.